Hello and welcome to our presentation of our research on comparison of stomatal activity in conditions of low and high intensity light in Zebrina plants. Our investigators on this research were Keila, Jennings, Eva Holder, and our principal investigator, Carson Huber. Okay, a little bit of background information on this topic. The overall equation for photosynthesis is the reaction between water and atmospheric carbon to form an organic carbon structure sugar and oxygen. So the first reactant of this equation is water. It is taken up through the root cells via osmosis. So there is a high concentration of ions in the root cells, which causes a chemical gradient and a lower water potential in the cells, which draws in water. The water is then transported to the location of photosynthesis, which is the leaf, by the cohesion and transpiration method. Cohesion is a property of the water molecule caused by its high polarity, so it interacts with other polar molecules, and that's how it is able to be drawn up the xylem, which is in the stalk of the plant. Transpiration also helps pull the water up the xylem. When water evaporates from the leaves, it causes a negative pressure in the xylem and pulls the water up. CO2 is the other reactant of this equation, and it is taken in through the stomata. Stomata are pores in the epidermal layer of leaves and are formed by two specialized epidermal cells called guard cells, which can open and close to regulate the movement of gases in and out of the cell. Stomata open in response to blue light. The blue light triggers the cells to actively transport ions of chlorine and potassium into the cell, which again causes a lower water potential inside the cell and causes water to flow in. The cells become turgid and open, and when there's no blue light, the ions are then transported out of the cell, and the cells become flaccid and open. Close. Plants prevent water loss in many different ways. One of this is stomatal regulation, so the opening and closing of stomata. Stomatal density, so the higher the stomatal density, the more pores there are for transpiration. So that is more of an evolutionary way that plants can prevent water loss, is by having less stomata. Sometimes plants also have a waxy exterior coating, which again prevents transpiration, and they can separate the carbon fixation and sugar production steps of the Calvin cycle. This is done in two different types of plants, one called the C3, which separate these two steps structurally, and it requires a second molecule or an additional molecule to move carbon dioxide throughout the cell, and CAM plants, which separate these two steps temporally, or they happen at separate times. And now we're going to go to Keila Jellings for our hypothesis and results.
Hello. So I'm here to restate our hypothesis and also show the results that we have gotten. So for our hypothesis, we created this through prior knowledge that we had and the information provided by our description. So to restate our hypothesis, if the exposure to sunlight of a plant is limited, then a higher percentage of stomata will be closed compared to the percent of stomata closed when the plant is in full sunlight or full high light exposure. So with this hypothesis in mind, we built our results, and I want to shift our eyes towards this table here that we have created, named the number of stomata open versus light intensity. Our independent variable is the high light intensity exposure and the low light intensity exposure. And you can see that there's photo number 1, photo number 2, and photo number 3. Those are our trials, perhaps we say, and each one is a set of photos from one in the one leaf that the lab group that we have gotten our data from has taken a picture of. And you can see here that the high light intensity exposure is a lot higher than the low light intensity exposure, and it's almost double the amount as the low light. Based off these 3 trials, we decided to make an average, which is shown here. Then we decided to create a chart, which is a bar graph that we have decided, because we really wanted to make sure that we compared the data. And time wasn't a factor in this, which allowed us to choose the bar graph instead of a line graph that you may see in a lot of common science experiments. And to our bottom right here, these are the types of photos that we analyzed, giving credits to our the lab group that took these photos. And through this, we counted the number of stomata open, and this is our results. And now I'm going to give it to Carson, who will be explaining to us our conclusion.
So I'm the PI, Carson Huber. I'll be talking about the conclusion of our lab. So as we can see in the bar graph, when the plant leaf is exposed to high light levels, there are more open stomata than there is on a leaf that has low light exposure. To prove this, we use the data from the group Yellow 8. They were an in-person lab, so as online students, we use their data to prove/disprove our hypothesis. So, and speaking of that, this data does not disprove our hypothesis that if the exposure to sunlight of a plant is limited, then a higher percentage of stomata will be closed compared to the percentage of stomata closed when the plant is in full sunlight. We theorize this because Zebrina is a C3 plant, like we learned in Bio 121. They have their stomata open during the daytime and closed during the nighttime. That is to regulate water loss more of using their guard cells. So this data proves, or supports at least, that Zebrina is in fact a C3 plant, which co-aligns with the data we have. For the limitations of the experiment, I believe that this experiment is very limited, but that is also mainly because we are an online group. So it was kind of hard to draw conclusions if we didn't have the physical data. There was only 3 plants for us to choose from in our data set, because we want to stay consistent with our data. And also we were not given the intensity of light, or the lux is the unit for light. So we weren't given the specific lux of the light, and we weren't seeing how far away the light was. So it was kind of hard to make quantitative comparisons for the 2 plants. We had to do a lot of qualitative stuff, which I think doesn't show a good analysis of the data. It's hard to analyze the data if it's qualitative instead of quantitative. So I think that those are the limitations of the experiment. That's it. Thanks for watching our lab slide, and please email us with questions if you have any.